Hey listeners, it's Paul Andriola here. Why not join our community at Small Cap Discoveries, where we offer our members direct access to some of the best microcap investment opportunities available. Our members are getting access to premium microcap financings, research reports, and direct access to management. Sign up today at www.smallcapdiscoveries.com.
Hi, everyone. Welcome to the Small Cap Discoveries conference call. Today in our call, we have the CEO, Jennifer Bath from ImmunoPrecise Antibodies. ImmunoPrecise trades on the NASDAQ and the TSX Venture Exchange under the symbol IPA. The company is currently trading at $7.10 Canadian, with roughly 23.5 million shares outstanding, or about $167 million market cap. I'd now like to hand it over to Paul Andriola.
Hey, thanks a lot, Trevor. It is my great pleasure to have Dr. Jennifer Bath with us today, of course, from ImmunoPrecise. Unfortunately, she couldn't get in front of a computer, so we are just going to hear her voice today, but I think that will accomplish what we'd like to today. So, Jennifer, welcome. Before we sort of jump into any sort of the questions, why don't you refresh everybody's memory? And certainly for those that are new to the story, tell everybody what ImmunoPrecise does and what they're all about.
Great. Thank you, Paul. And thanks, Trevor, for the introduction. And I'd be more than happy to give a background here, especially as IPA just continues to grow and continues to change, and really help put this background also for those of you who have been following IPA, into some of the context strategically about where we have been going and some of the reasons why we've done some of the things. I think some of our strategy now will make a lot more sense to you after this recent acquisition. And so I'd like to, revisit that background story with a little bit more of that language about where we have been going. And hopefully it will just be really clear to people what our vision has been and how we're able to bring that forward to you in a new light today. So first of all, I'll start off for those of you also who may not have much background in IPA or our story. IPA is a contract research organization, so that's better known as a CRO. And what that means is we're hired by pharma companies to discover and develop new antibody therapies to treat diseases. So what we are known for at IPA is our robust technologies and also our scientific rigor. So industry leaders out there are committing to IPA in part due to our ability to customize challenging programs and to ensure the best research and clinical outcomes. So that customization that we have at IPA, it really is unlike what you see at most contract research organizations. We don't apply what we refer to as a one-size-fits-all approach to treating diseases. We have decades of experience that have taught our scientific leaders throughout Canada, the US, and Europe, not only about the importance and the understanding of how to treat a disease as it progresses, but also how specifically we can alter the course of that disease. And also what antibody formats are best suited for a particular therapy. And one of the things also that we're quite knowledgeable in is understanding the characteristics that those antibody therapies really need to possess. So while most companies out there, they'll onboard like a task coming in from a pharma company of developing a new therapy, and then they'll just push it through one or two different discovery protocols, we are always innovating. We're bringing forward the best-in-class technologies, developing them, acquiring them, whatever it takes, to really ensure that we have top-notch technologies to ensure that the outcomes for our clients and our partners are something that they can be proud of. And this enables us to design a custom plan, which is really what's key about what we do, to maximize the quality of the output, wherein our final pool of therapeutics that goes to the pharmaceutical developer contains really the most meaningful, the most diverse, which is a word you'll hear us talk about frequently, and the most relevant clinical candidates. So for those of you also not super familiar with our capabilities and or listening more recently to our last quarterly earnings call, we did touch on what has really become one of our most popular platforms, And that's our B cell select technology, utilizing what we have coined as a function first approach. Our B cell select technology is one that enables us to screen up to 10 million immune cells per run. So what we do is we go straight to the individually isolated cells, and we determine which of them are producing antibodies with characteristics that make them suitable drug candidates. And then we go straight to the DNA sequence that encodes that antibody. And that gives us the genetic code that we need to reproduce that antibody as a drug in the lab. And then we can test it as a potential therapy. So where we excel here is not just in the number of B cells we can reliably screen or the amount of information we can collect about each antibody prior to getting that genetic sequence, but it's actually in the fact that we can also deduce the exact function of each antibody prior to selecting the therapies. So I know we mentioned this in our last quarterly earnings call, and transcripts are available for that, but I want to put this in some context for the listeners so that it's clear exactly what we mean by that function first and exactly how it helps us select the best therapies going forward, because it really is, it's being adopted widely for a reason. And that's because it really does change the game for antibody developers at pharmaceutical companies. At the end of the process, instead of turning over hundreds of genetic sequences to pharma developers who then need to determine, all right, I have all these sequences, which one encode a meaningful antibody therapy? We test the individual antibodies produced by the cells to determine exactly what their function is. For instance, are they performing in a way that may reduce or eliminate disease? To provide the pharma companies with only the most meaningful candidates to move forward, saving them time and energy and obviously also money. So when we talk about an antibody function, we say that, we're only selecting candidates that have a particular function. What we're really referring to is the ability of that therapy to carry out a very specific activity to either treat or even cure a disease. So a good example of that would be like in an oncology study. Researchers can test the ability of an antibody to block cell growth by determining, is this monoclonal antibody blocking the connection between the cancer cell and the proteins that promote cell growth, which would be considered an activity necessary for cancer growth and survival. When we perform a functional test such as this, we would select all the antibodies with that particular capability of blocking the binding of the proteins. And we may even test for the lack of the cancer cell division and cancer cell growth. And then we immediately remove the majority of ineffective drug candidates from the development process so that the pharma researchers can focus their efforts only on the subset of the most promising therapies. And that's the power, the real power behind what we do at IPA's Function First platform.
Jennifer, I don't usually interrupt when everybody's describing, but one of the things I really want to make clear is that what you're really saying is that the sort of the benefit the client gets here is that not only do you find sort of the antibody or the therapy, but you're sort of characterizing it or giving as many details as you can so that they have significantly less risk as they sort of advance it forward into the different phases of trials. Is that correct?
That's absolutely right, Paul. So, one way to look at it as the majority of companies out there are first and foremost, providing solutions that bring forward a minimal number of candidates in terms of how unique each of them is. And when they turn them over, it usually just the answer is typically, does it bind or does it not bind to a target? And then there are companies who provide slightly more high throughput processes who can also give the genetic sequences of the antibody. However, the majority of the time, those companies are just handing over hundreds of sequences and there's no meaningful information about, does that sequence encode for something that binds strong, that has the function you're looking for? Does it stop that cancer cell from dividing? Any of those questions that would be go, no-go decisions for those lead candidates. And so, you're really just kind of it's kind of a minimal effort and typically a low output with in terms of the amount of information that really leaves clients and partners kind of on their own to go figure out what is actually meaningful about the data you've turned back. And we're able to provide them with all of that work with great diversity in the results, and also in a fraction of the time that it typically takes to get much less meaningful results.
And we call it more shots on net up here in Canada.
Yep. I love that analogy, especially as a soccer player.
Exactly. Well,
Perfect.
hockey for us, but yeah. No, and clearly, a question I usually get is, why wouldn't, the big Pfizers of the world or the big pharmaceutical companies, why are they not doing this in-house? Why are they hiring a company like IPA to do this for them?
Yeah, so that's a great question too. So, historically, a lot of pharma companies did try to develop the different kind of cutting edge technologies in-house. And that actually, we started to see a pretty major change in that about 10 years ago, where what was happening is that the types of targets that people were going after were changing. They were getting much broader. They were getting much more sophisticated. And so the number of people that these companies needed to employ with the different skill sets, and then the number of technologies and the amount of equipment they needed to onboard was incredibly vast. And they might sometimes only be for, one or two different targets in a particular disease class. And so it didn't enable them to switch directions quickly, to be very nimble, or to really go after a broad class of diseases. So they started seeing a large push for these pharma companies to outsource to companies like IPA or contract research organizations. And today, the vast majority of pharmaceutical companies continue to do that for those exact same reasons. Ultimately, they can move more quickly, they can address a broader range of targets, and then they can tap into specialized personnel who know exactly, how to do that drug discovery on that type of target on that platform, and have done it for decades without having to, onboard all of those different skill sets. So it's been a win-win in a lot of ways, with the exception that, contract research organizations have really over the course of time kind of failed to be truly innovative. And that was one of the dearths that we saw in this market. People were just doing really, by and large, what the founder of a company learned to do in grad school and did well. And then they spun out a little business. And we had hundreds of companies come into this market where they did, one or two things. And that became the philosophy, was that kind of one size fits all, instead of really mastering and then investing in keeping innovative technologies, building and growing, and staying ahead of what was needed in the industry. And so today it's really, at this point, there's still the vast majority of the work is outsourced, but the challenge is for those companies to really find CROs who not only have the experience, but have the technologies that can get the job done in a shorter period of time with really meaningful output.
Yeah, we talked a little bit about shots on net earlier, but really, I think what distinguishes IPA in a lot of ways is, we've got more than one or two discovery platforms. Maybe speak to that a little bit, and why that's unique, and why that's an advantage for the company.
Absolutely. Our philosophy, kind of goes back to this recognition that these pharma companies were really being asked to outsource the majority of their work. And one of the ways that IPA responded to that as part of our major cornerstone of a strategic plan back in 2016 was to bring, all of these a suite of services, end-to-end services, in-house. Leading technologies, complementary technologies, but also only those technologies that really, really brought forward the best of the best in the services, so that these could all be performed at one company, and really reduce the risk of these pharmaceutical companies having to cobble together a dozen or more companies. And so that is definitely true in terms of where we really put our efforts was in that discovery platform first, because the truth is that's really the funnel that's going to dictate the quality of everything else you do. So, we have this B-cell select platform that I mentioned to you, which is probably our most popular platform right now. And part of the reason for that is the function first I mentioned, but also the fact that it is species agnostic. So if you're going after like an oncology target, for instance, it's a target that originates in the human body. So it's really hard to get human antibodies to recognize a human target. The whole point of how the immune system develops is to prevent you from recognizing your own proteins, right? Otherwise you have an autoimmune disease, right? So the further, away from the human proteome, or in other words, the more we can use animals that are less like a human in terms of their genetics, the more diverse antibodies we're going to have, which basically means we're going to have antibodies with way more functions. And it's those functions that dictate how good they're going to be. And so that's one of the reasons that platform is so popular, is it's species agnostic. We can really use any type of species on that platform, which is almost unheard of. And the vast majority of companies are relegated to using rodents whose genetics are very similar to ours. Now, that being said, we also have phage display platforms. We actually acquired a company back in 2018 called BoniQuest Research, specifically because they had an awesome reputation for their work in phage display. And they also brought to the company a lot of downstream capabilities that are important as you're preparing a final product for the clinic. But those capabilities are quite interesting as well. They not only have human phage display libraries, so a product is human coming out of even the initial discovery phase, but they have camelid libraries, so they can make what's called a VHH antibody. We have what's called an SZFV or fragment antibodies that can come out of our phage display libraries. So we basically can build antibodies of all different formats with that discovery platform, and also from multiple species. So antibodies that are a single chain, antibodies that are bispecific, antibodies that are trispecific, like small portions of antibodies. All of these have different reasons why you might want to use one in a particular situation over another. And that starts to boil down to, our collective 65 years of experience in knowing which type to use and then how to engineer it properly. And then we also have Hybridoma Technologies, in particular Hybridoma Technologies coupled with a immunization platform, which takes 19 to 21 days to immunize compared to a traditional 60 to 80 days. And then our Hybridoma platform is what we call a single step platform. It eliminates what other companies do, which is months of subcloning. And from the time someone brings in an idea, for us to perform on that platform, to the time we, immunize the animals and have a final product ready for them is about 30 days. And that's compared to a traditional 9 to 10 months. So there's different reasons why people love different platforms. And those are some of the discovery platforms. And we've really worked over the years through R&D and through acquisition to make sure that at every step of what we do, whether it's building an immunogen, whether it's immunizing an animal, whether it's the discovery platform, whether it's how we characterize them to really get the ones that are going to translate best in the clinic. Every step of what we do, we've really worked to bring forward a multitude of options, knowing that different options work better in different situations. And they all enable us to customize programs using our experience, knowing typically what is really going to provide a candidate that is most likely to be successful in the clinic.
The way, so, we always like to phrase it sort of, what problem do you solve for your clients? And here, correct me if I'm wrong, what you do for your clients, which is typically anything from call it a small biotech all the way up to large pharmaceutical companies, is you give them the most options available for a specific sort of problem, and you give them the most information around those options, right? So it's as many sort of candidates as you can find, but you're also giving them as much information as those candidates, so that when they go forward, they either lose or fail as quick as possible, or you're increasing their chance of seeing that success going through the phases. Is that correct?
Yeah, absolutely. Yeah. That's a huge part of it. And I think one thing to add to that is that, I really think clients appreciate, I know they do appreciate that customization and the breadth of what we're able to do. However, most of them do not want to be handed a menu They don't want 1,000 service offerings. Well, we literally have hundreds in the course of a whole program. And so what they really love is that we have all that diversity of options and these tremendous platforms and this ability to customize, but we have the scientific know-how to put together these options in a fashion where we say, this would be best because of this reason. And then this, yes. And so we're really taking a lot of the guesswork out of developing new molecules for our clients.
Yeah, no, well said. Taking a step back, what do you see as some of the biggest challenges, developing cures for patients, such as cancer, and why is it so difficult? Why are we not finding the cures for cancers easier than we are right now? Yeah,
it's a good question, and it's a question I get a lot because I think a lot of people are frustrated by the lack of progress that we tend to see in oncology therapy. And that makes sense. We've been struggling to treat cancer for a really long time. But it's a great question because it ties in exactly, where we've been and the challenges that we saw. It's not just challenges for people to discover good medicines. It's the whole process by which we go about, discovering new medicines and treating patients. And I think that's a part of where our strategy is really starting to kick in. It's a problem we've seen for a long time, and it's a problem we've wanted to address. And that is a big reason why we also have been looking at companies like BioStrand. So if I had to pinpoint what I thought the biggest challenge was today, I would say it's developing therapies that are broadly applicable to a multitude of patients. And if we look at that from a patient side, there's just a lot of limitations and very significant limitations with the therapies we have. And I think cancer is actually the best example of that problem that we have. The issue is, like, current drugs that we have to treat diseases like cancer, they don't work for everyone. And so they're super unpredictable when they're prescribed to a patient. And they're typically prescribed based on the outcome of a clinical trial where, they were selected based on determinants that are completely unrelated typically to the root cause of what the patient's cancer is. And so I think one thing that we've just tended to forget, or actually maybe more realistically have not been able to address in science today, is that everyone's genome is different. Everyone's genetics are different, right? And everyone's susceptible to different diseases. And when someone gets one of those diseases, they're going to respond very differently to each therapy. And so for treatments to really be effective, they need to be selected specifically. And I think this is the important part. It's specifically for the mutation that caused the disease in the person. So if we look at an example, which I think sometimes just makes it, easier to kind of digest all the science and the challenges we have in medicine. If you were diagnosed with cancer today, the odds are strongly against you being prescribed the right therapy. So a therapy that matches the exact mutation in the protein that gave rise to your cancer. And the reality is that most of the time, the first chemotherapy that's selected is not going to work for you. And it's kind of grotesquely referred to in medicine as the cut, burn, and poison approach to treating cancer. And the reality is, everyone's disease is unique and they shouldn't be treated the same. And it's not a personalized plan that addresses the actual disease the patient has. It's just a broadly applied plan, approach where we treat patients when the exact root cause of the cancer isn't known. And if you don't mind me elaborating on that a little bit, I just think this is kind of fascinating, Paul, because how we get to that point goes all the way back to how CROs are developing the drugs, and then how they're actually being tested in the clinic. And so, when we run clinical trials, oftentimes they're being run on well, if you could see me, you'd see me do little quotation marks with my fingers, because I always refer to it as the type of cancer the patient's been diagnosed with. And so a trial might be testing a drug for patients who have all been diagnosed with, again, quote unquote, colon cancer. So the major challenge in cancer therapies today is the fact that there's no such thing as colon cancer or
Mhm.
liver cancer, right? Or brain cancer. Instead, it's a multitude of different types of cancers. They arise because there was a specific mutation that changed the structure and the function of a very essential protein, right? It
Mhm.
goes right back to a mutation. And so, the colon just happens to be the organ that the cancer is growing in. And the actual disease itself
Mhm.
is based on the mutation. And so to develop a really meaningful drug, it means you need to target specifically against the mutated protein. And, that's where we go wrong today, around,
Yep.
I think I read it was like over 95% of patients will be put on the standard of care. And if that stops working, well, you hope the second option works, or if
Right.
not, they may not make it. And so that's why we really, we need to have an informed, contract research development. We need to know exactly what the mutation is. We need to understand the structure of the target. These are all things that we've been quite good at IPA. And then, be thoughtful about how we develop specific therapies from a broad selection of antibodies with a determined function. And that's very essential to, being able to very precisely treat a patient and also have better outcomes and fewer side effects.
Mhm. You used the word personalized, and we're going towards this concept I was going to bring up later about personalized medicine. That's really what we're talking about here, is understanding the human, understanding what the illness really is, what other issues that human might have, and therefore, personalizing that therapy or course of action. That's really what we're talking about, and we're going to get into that a little bit more, because
Okay.
I mean, really what IPA has been able to do is through its different discovery methods, the amount of data that you guys have that is applicable to personalized medicine. And of course, with BioStrand, very anxious to sort of hear a little bit about that. But even before we jump into that, I keep hearing the term Omics. Please let us know what Omics means and why is that important?
Yeah. Well, I know we've been using that word ourselves quite a bit lately, and it's so omics. First of all, it's really the suffix that we add to basically the study of large amounts of data from different sources, including, so for instance, we have genomics, which is where field of study where we measure DNA. We have transcriptomics, which deals with RNA, just from the root of a transcript, RNA transcript. We have proteomics, which is obvious, right, for protein, and then metabolomics for metabolites. And the reason why, we have been starting to introduce these concepts to individuals is that, the omic data analysis is actually typically the starting point for the very first step in developing a new drug. And so that starting point is target identification. So a lot of our clients at IPA, they actually start with, before they come to us, right? Like long before they come to us, they oftentimes are starting with like large amounts of clinical trial data, omic data, whether it's DNA data, RNA data, protein data, large amounts of omic data, from both healthy patients and diseased patients. And then they use these high throughput omic approaches to generate literally like tera to petabyte size data files. So massive amounts of data. And with these data file sizes, like together with differences in nomenclature and different types of data, and then, taking all of that information and being able to like integrate it into data that's actually meaningful, right? When you want to select a target, and interesting and meaningful in the biological context is where we've really struggled historically. And so this is really true if you want to understand like a biological function and then make correlations to a disease and how to treat it. And so the ultimate goal that researchers has is, you want to determine exactly what genetic mutations exist, right, in the patients. And then what is the impact of that on the protein and its function? And then how can I use that information to develop an effective and safe therapy so that I can exploit that, mutation in the structure? And as I mentioned, like a lot of times biotech or pharma clients, they come to us with a target in mind, but usually they've spent years analyzing massive omic data sets, right? Trying to get some sort of usable information. And then after analyzing those data sets, they typically have to turn to a lot of what we call unstructured data sources. So primary literature, patent databases, try to understand like what is that mutated protein specifically? What is its role in the disease? Why does it give rise to, for instance, this particular type of cancer? What research has already been done? What's the viability of the patent landscape? You name it. There's a ton of research to be done.
Right.
And all of those research areas are incorporated into BioStrand's Lens AI software. So what would normally require months or even years of research and enabling access to relevant data, or normally like months and years of research out there using multiple software programs. I should also, say, because you can't analyze typically genomic data on the same software as transcriptomic data, et cetera. Now, what we're doing is enabling access to the relevant data using Lens AI in seconds or minutes, as opposed to months or years. And we can talk a little bit about how that's done and why we're able to search so quickly, but it comes back to the hips, which I know several people are interested in learning more about.
Yeah.
And go ahead.
And I was going to say, I mean, you're diving right into BioStrand, and before we get too ahead of ourselves, why don't you just announced a 20 million euro acquisition of BioStrand, which is a company based in Belgium. But try to tell us exactly what is BioStrand, not so much what they do, but I mean, a small organization. This is a software company, really. But tell us what you can about BioStrand, and we'll get into sort of the problems they solve and some of the future possibilities of problem solving from them.
Okay. So in terms of like, what is BioStrand? You want to know about the technologies, or
well, let's dive into
revenue,
type of company. As much as you can tell us. Is it 100 people working away on computers? Is it what is BioStrand?
So BioStrand is a relatively small, primarily family-owned company. They have somewhere between 12 to 13 employees. We have a little shuffling going on right now. We're adding some additional positions on top of that too. And it's run actually by a husband and wife team. And their names are Ingrid Brands and Dirk van Hifta. And both Ingrid and Dirk, the founders, are MD PhDs. They're incredibly brilliant. But what I actually love most about the way their minds work is they think outside the box. To them, there are no limits, and they see possibility where other people, especially with very black and white or traditional training, would be very hard pressed to step outside of the line and realize, what they're actually looking at. And what I think is actually interesting here is they were actually practicing clinicians by trade for many years. And they ended up leaving their previous careers when they realized the power behind the codes that utilize the HIFs and what we call the multi-level network that they create. And so the company itself is made up mostly of data analysts and coders. And that is primarily, what they do is code software. And the software has accessibility to not just therapeutic antibody discovery. That is what they will be focusing on with us, but it has relevancy to anything in the biosphere that uses a genetic code. So vaccine development, target development, clinical trial design, clinical trial output data analysis, obviously omic data analysis, agriculture, you name it, there's a lot of different applications.
Basically, the way I see it is they have the ability to basically analyze almost any living cell. Exactly, right? So you even mentioned agriculture. I mean, that's a far step away from antibody therapeutics. So the breadth of what can be sort of worked on here is very wide.
It is really wide. Although one thing that I think is maybe one of the coolest things about this, Paul, is that we tend to put unnecessary filters on our perspective of what we need to include when we do drug discovery, right? So it's like, yes, anything with a sequence out there in the biosphere is relevant, but the reality is, like, when we do even, antibody drug discovery, we need to take into account all sorts of information. Again, their software can manage DNA, RNA, and protein information all on one software platform. And there is no other software that I know of in the world that can do that. But it's going to take it all and it's going to reduce it to the most common denominator, which sounds like a simplification. It's not. It's actually incredibly complicated, but these are those hip fingerprints. But in the meantime, because it's narrowing it down to that, and you're not searching using these algorithms with entire sequences, you're searching with the key fingerprints, which relate not just to sequences, but to function, to structure, to very meaningful evolutionary portions of sequence data out there. If I'm analyzing, for a particular antibody therapy to use in a human, I'm going to get back data not just about the different antibodies or maybe the target of interest. I'm going to get information about all other targets that might actually be present in the human system that could be an unwanted effect, right? And it does that so incredibly quickly, again, because you're not searching tens of thousands or hundreds of thousands of sequences. You're just deducing to a finite number of fingerprints that are known, that are indexed in the coding, that allow you to push that information incredibly rapidly and get it back. And it's also going to give me information back when I'm developing a new drug about the impact that drug development has on my microbiome. So all the gut flora that live in my intestines, they impact my immune system. They impact what antibodies I do and don't make, and what drugs might be most efficacious for me personally. All of that data is coming back. So anyway, I could go on, but yeah.
And I want to try to simplify, because I can tell you're pretty excited about it. So really what this does is it first off, you mentioned HIFs. They've categorized and indexed, I think it's roughly 660 million HIFs. That's a finite number. And the ability to sort of be able to sift through that and look for certain data that you're looking for is very unique, my understanding, very powerful. And it's at the speed of almost a click of a button. So, it's a powerful system, but how does a client use it? Like if I were a pharmaceutical company, or if I'm one of your clients, what happens? I walk in the door, or I give you a phone call, and I say, what would I say? Here's my problem, and you guys tell me how you're going to fix it. I keep coming back to fixing problems. Simplify it as much as you can.
That's what we're here to do.
Yeah, simplify as much as you can.
Yeah, I absolutely will. And the short answer is that, people come with all different sorts of problems, and we have different ways to solve them. But some of the most common ways are, that first example of, oh my gosh, I'm drowning in omic data. How do I manage this? How do I take something meaningful out of here? And there's a few ways they can use BioStrand to do that. One is they can actually take a license to the software, and they can license that software based on the number of users that need to access that software. And so for larger pharma, that's oftentimes what we see in this industry. They'll be, accessing a large number of seats for software licenses to be able to, interpret and analyze data on a daily basis and make key decisions. And really all companies do that. We access and license several software programs ourselves already prior to purchasing BioStrand, because commercial entities cannot use public databases for client data or their own data. They're not secure. And so they're very common. So, people can do that and plug in and access and connect to the BioStrand software to analyze data from that perspective. However, what we've been seeing here, as we've been kind of, reading reviews and talking to some of the prospective clients for BioStrand, and also getting feedback from BioStrand on recent meetings. For instance, they attended Bio Europe, as did we the week before the closing of the acquisition. And what we're finding often is that larger pharma are also saying, yeah, that's awesome that we can plug in, and that software can do things that, no other software can do. And we've heard that from other pharma clients too. Like, it's tremendous what it's capable of doing. However, can we actually connect and give you all of our information, and you can analyze it for us? So we can also offer it as a service. And I think there's some strong benefits there too for companies who are primarily biological based in terms of their research, and don't have deep bioinformaticists on staff that would be able to put that time and energy into that research. And so either one of those can be done when it comes to the omic data analysis. Now, when it comes to some of the offerings that we can already offer through BioStrand right now, and this is something we've already been doing. We have some early adopter clients that we've already talked to about some of the offerings at BioStrand, and early adopter clients who came in and said, hey, this is a problem like super specific to this program I'm working on with IPA. Can we try and see if LensAI can solve this? And in those cases, that's kind of like a CRO fee for service setting, where they want to come in and say, hey, I have all these drug candidates. Can you run them through your software and first make sure I didn't miss any? And second of all, see if there's anything predictive about the function of other antibodies out there that I might be able to add to my repertoire, maybe expand the number of lead candidates coming through. And, or can you maybe tell me a little bit about whether or not these are likely to have off-target effects, which means like if I give it to a patient, is it going to bind to that mutated protein that's helping cause cancer alone, or is it going to also bind to, a protein on a tissue that's gonna cause some toxicity? And all of that can already be done at BioStrand. So that kind of fee for service application is a way for us to expand our offerings in a way that's very powerful and not readily available at most CROs. But then, as I said, there's other ways for them. There's ways where people can come in and custom and say, hey, this is my problem, solve it with LensAI. Or, again, having us do the research for them, which is purely like on the computational side, or take a license. And then I don't want to leave out that whole aspect of, for Talem Therapeutics, which is our subsidiary and affiliate that does its own drug discovery. Talem works on high value therapeutic antibody programs and has already done a couple of projects with BioStrand during the process of the due diligence work. And, that's a huge opportunity for Talem Therapeutics to be able to utilize this software to further give them an edge in the development of these assets. And there's some really interesting kind of changes in the thought process of people's perspective of how AI and machine learning is actually now impacting the value of preclinical assets, which is the mission of Talem, and how it actually brings like the net value, or may bring in the near future, the net value of preclinical assets much closer to what we consider the net value of a clinical program to be, just based on the enhancements we can gain through AI. So correct me if I'm wrong. What I just heard is that when it comes to Talem, what you're able to do is sort of advance these Talem assets by sort of applying AI and getting a lot more data that in some cases you probably would have had to wait to see in human trials or later stages. Is that what we're saying? Absolutely. And also offer it to partners within Talem, who are really interested in, So there's a lot of different ways people can access and will start accessing quite shortly here, the BioStrand offerings. Gotcha. Now, the question and sort of the common sense thing I would think here is that you have a small company like BioStrand that's sort of working away, toiling away, sort of in their laboratories. They don't necessarily have the same access to clients like you do as a CRO. And now, does this move that offering much closer to the client base that you guys have? It should be easier, if anything, to be able to go to one of your existing clients and say, hey, try out this new service along with everything else we've got. Is that not the case here? That's absolutely the case. And that we saw that as a huge win there, because BioStrand, first, they've been so focused on the coding. They haven't really been focused on the user interface, and they have not really been terribly focused on getting those clients in the door. But the ones that have given feedback A lot of them have said, first, one minor criticism is like, there's so many different ways to attack this and approach it. We don't know what to start. Help us, right? And that's not a surprise. And then, one of the kind of threads, like a common theme we've seen throughout there is, oh my gosh, this is amazing compared to these large software programs that we're able to access and bring in. However, this company is so small. And is it going to be here in a year or two? Because that's a massive investment to make in a company that, may not be there later. And that's an area where we obviously come in and provide a ton of support and stability and security. But yes, accessing, being there in Belgium, they've kind of more recently just started getting out and telling their story. And they're also almost like too smart for their own good, because they are deep in the omics and in the data analytics and the bioinformatics, and telling the story to like lab scientists directing programs. It has really been challenging for them, and to really translate into what the software means for them, because the ones heading up the programs are not always the ones doing the large scale omic data analysis. That's, you've got to meet two separate groups there, and they've got to both understand the power of the capabilities. And so yeah, it's, for them now to be able to access the clients that we have, we've already seen that interest, like I mentioned, coming in from our early adopters that typically grab onto our early technologies. We already see some bringing really cool ideas forward. And now we are, we've spent the last 3 weeks or so integrating their offerings into our marketing, brochures, capabilities, exhibition materials for conferences. We've put together different case studies of how when our existing clients are working with us, they can tap into all these extra capabilities at BioStrand. But maybe one of also the more cool things that we're really looking forward to is that on May 2nd, where we'll be heading out to one of our larger conferences of the year called PEGS. It's Protein Engineering in Boston. We have an event set up there where it is going to be limited access. It will be an annual event. But we will be really presenting some very key things from our contract research, including the function first, which deals with a lot of data associated with function, but also as a way of tying very nicely into BioStrand's capabilities. And our keynote speaker that night will actually be one of the members of BioStrand's management team. And so on May 2nd, we will be doing our large unveiling in Boston at PEGS. And then we have a double exhibition booth, just this kind of like really in your face, massive, but beautiful exhibition that goes through all the integrated offerings and how omics and data analysis changes everything these clients do before they even figure out what they want to work on, before they walk in our door, and how now we can back up several years in bringing them through this process in a very meaningful way into the CRO door and really help them from beginning to end. And so yeah, that access to those clients, really integrating it into a meaningful workflow and helping them understand, wow, how this changes the game, how it gives them meaningful data, how it really changes the scope of the analysis and what they're able to do in the laboratory.
And I got to imagine the relationships you have with existing clients. There's probably problems that BioStrand didn't even know existed, and you don't know until you get feedback and conversations going with clients who say, "Hey, I've got this problem over here. Do you think you can help us?"
Yep, that's exactly, and it's exactly right. And we've had a couple already where, a lot of the software out there and a lot of the way people try to target problems today is based on sequences. And the reality is you need the structure, and from structure comes function. And so, we have a lot of clients who have dealt with key core issues in developing therapies because they don't truly understand what the structure is and how that's impacting the disease. And so we have a few of those right now, and we, and a couple of those in particular stepped up and said, we're working on this, and it's going to be a really, really challenging thing for us to solve. I don't want to give the details of it because it's kind of, specific to a particular company, but absolutely, when they heard about the software, even at kind of a high level initially, it was, oh my gosh, I have a feeling that, that software is going to give me a whole new perspective on how to address this. And their plan is to move forward with that. And so absolutely, it's a huge value add for our clients. And then obviously for us, where our main goal has been, let's help them every step of the way. But going back to what we've always said, when we help them, we have to bring the best tools forward. We don't want someone to be with us just because we offer everything. We want them to be with us because we're the best.
Exactly. You want to solve their problems. Exactly. Now, here a question I should have asked earlier, like, how did you find BioStrand?
That's, I've had that question like 4 or 5 times this week alone. And so, we, and that question, and also people wondering, like, how did you get BioStrand instead of, a larger pharma group? And I think they were the best kept secret, really. Although, to be honest, there are a couple of other groups that we do know that we're looking at BioStrand. But it kind of tapped into some of the greater concerns or fears that the founders had, which was they might be stripped of their intellectual, freedoms to really bring this to fruition, to create new drugs, and instead get swept into a larger technology company, and which might have ultimately been their fate, right? A genome sequencing company or even a computer company would have great use for this software. And so the way that we found them was, we actually have several internal M&A teams at ImmunoPrecise, and they're classified based on like targeted areas of specialty. So we have one team that specializes in AI, machine learning, and omic data analysis. And that team has been working for about 24 months to scope out potential M&A targets. And then they report back to the rest of the executive team on things like changes in the competitive landscape. And they work with the scientific team to build out things like internal structures and services and IPA, so we can align with different transformative capabilities that they've uncovered, and really prepare to integrate eventually into a large data AI or some sort of algorithm coding. And so this team, they actually, quite some time ago, they initiated pilot projects and collaborative programs with the lead companies that they had selected as the primary M&A candidates. And that enabled us to review output products from each of those candidate target M&A groups. And we were able to get through those outputs from those companies by I think it was mid-October of last year. And that was kind of the final part of our early stage due diligence, really get in and better understand the power, the capabilities, and the potential for all of the different algorithms, machine learning and AI, and what was truly transforming drug discovery and what was just kind of a basic incremental step, which I think we see a lot of, especially in more kind of promotional language about, AI and machine learning, because they're just kind of like sexy phrases people like to throw around, quite frankly. And then with BioStrand in particular, we went in and we conducted one really extensive study and then a couple of smaller studies. So we had a case study that analyzed, this was actually really one of the cooler things I've experienced in my life. It analyzed a particular target, happens to be the specialty of a member of our management team. And so they know this asset inside and out, backward and forward, this particular target. And we collected data on the BioStrand platform from this protein and got data back really for things. It pinpointed things in the research and in the literature that were results that were immediately identifying as potentially meaningful, as potentially effective drug targets, but data points that never in 20 years of research have ever been pinpointed or selected as meaningful as a part of this target, which was mind blowing. And then we also did a much larger study where it revealed hidden but really interesting, important correlations between the human proteome, the gut microbiome, and then also susceptibility to certain diseases. And I think what was really intriguing about that was also quite a big aha moment for a lot of our M&A team was the realization that there was no other software and likely no amount of time and research that would have ever brought together those specific data points and provided us with such tangible information on like the path forward that we would go and what a potential outcome would be. And so those were kind of in that search. And I want to say we analyzed somewhere between 5 to 7 different software companies or companies that to some extent do software analytics coding. And, we narrowed down to 3 or 4 eventually, and then, completed these studies around last October. But it was really about the Lens AI. And for us, ultimately, that was the core of everything and how it alters our conversation about what is meaningful in computational analytics, what is meaningful in AI. And for the first time ever, changing like that narrative that, quite frankly, I'm a little tired of hearing after so many years of us. That deep due diligence for M&A was about 2 years, but it's been 4 years since we've been scouring this landscape and talking to these companies. And the verbiage ultimately on that you hear behind closed doors, it's all the same. It's about being able to use brute force to analyze massive amounts of data, and the same for like Google's DeepMind software. It's not about that, and that's what we loved about this. It changes the story. It's not about how much data we can analyze and how much data we can push through and how powerful our tools are for analyzing and how much storage we have. And we don't need to eventually go to quantum computing. It's about intelligent coding that provides rapid access to super meaningful data that's actionable and that actually can transform drug discovery today. And for us, that was end of story. BioStrand won that search hands down.
Wow. I want to go back to something you said, because that blew me away. So you mentioned a target that somebody at IPA knows fairly well, that data was found by BioStrand that was never found in the 20 years of sort of research on that target. So how powerful is that if you can do that with all the different targets that are out there? And wow. Okay. So that I think that sort of, it does sort of reemphasize some of the power that this thing has. Now, we use the word HIFT a couple of times, and we really properly described what a HIFT is. Maybe let's try one more time to help people understand what a HIFT is and why that's important.
Yeah, and so it's I'll describe what it is, but I always fear that in describing what a HIFT is, that it's going to minimize people's understanding of what's really significant about them. Because there's what is a HIFT, and then it's like, what is a HIFT in the broader context? So when we talk about, the genetic language and the syntax of biology, and then, and then there's all the aspect about what does it mean to be able to code a HIFT? Like a HIFT is a fingerprint, right? It's a genetic fingerprint in a sequence. But what's upstream of the HIFT and downstream of the HIFT, and what HIFTs can follow which HIFTs and what parts of sequences and what cells and what tissues and everything, that's all super important too. And all of that information had to be taken and coded into a program. And so it involves so much more than just physics or biochemistry or an understanding or identification of a sequence. But I'm going to give it a go.
Keep it simple.
I just don't want people over.
If you had a piece of paper and crayon, that would be the only tool you have to describe it.
Okay, I'll give it my best shot. So, I mentioned it's a genetic fingerprint, which means it's something easily identifiable. It's unique. It's relatively small. We can get in there and find it right away. Now, what's really important about that HIFT sequence is it corresponds to a structure as well. So that HIFT fingerprint isn't just a code. It has a corresponding structure, and that structure has a corresponding function. And the reason why you care about that is those functions are what cause disease. It's not the genetic sequence, right? And so, if you're if you're, quite frankly, to another interesting part of this is if you have a mutation that's causing a disease like cancer, it's probably a mutation in a HIFT. And it probably converted the function, and that function is now the wrong function and it's giving rise to a disease. And so hence, I think it could be, again, like really oversimplified, but the fact is, as you mentioned previously, you know, there's a finite number of sequences in the genetic code. And, you know, after years of identifying and coding those fingerprints, you know, by Dirk and Ingrid, one thing that was really clear is that finite number at like 660 million HIFs, they're universal. They are consistent throughout the biosphere, and as they represent that most meaningful data in drug discovery, in the context of whether you're working with RNA or DNA or protein, they are in there, and they are the most meaningful part that translates to, you know, the quality of your data discovery program. And because you can take those HIFs and you can code them into software, that's really what transforms the drug discovery and the conversation about what's important in AI. Because normally in analysis, if you end up with a bunch of DNA sequences, for instance, and you want to analyze those DNA sequences, you're going to need to push those DNA sequences through algorithms where the entirety of those DNA sequences are going to be analyzed, and the majority of that information is useless. It's not important, but that's a ton of data to process, and it increases exponentially every time you add a new DNA sequence, as opposed to being able to code software based on the HIFs, and then being able to take those sequences and be able to map the fingerprints, and in what we refer to as a Google-like indexing, as opposed to pushing with brute force. What happens is when you put your sequence data in, it just quickly searches through it all and gets rid of everything that's not meaningful. And it deduces it to the HIFs that are present, and then pushes that data through indexed data in the software to be able to eliminate the majority of the noise, if you will, to pull back in a very, very rapid fashion only the information that is most important. And so a large part of that is due to the fact that we reduce to the HIFs, but a large part of it is also based on the way the software is coded and the way that information is indexed, and the fact that we're getting, you know, directly at what we're referring to as the syntax of biology.
Jennifer, I mean, I keep sort of hearing these things that make it sound like you're saving somebody a lot of time. Can you sort of quantify in any way the time savings that this software, you know, produces?
Yeah, it's you know, I think that the biggest time saver that we're able to offer right now off the shelf is the omic analysis, you know, early on. So if you're starting with, you know, tens of thousands of data sequences, or quite frankly, if you're doing, you know, if you're using like small NGS sequences, we could be dealing with billions of sequences. But if you're dealing with massive amounts of data, you know, the ability to analyze any type of data on one software program, and then to be able to pull back not only that structured data, so that's the sequences you're pushing information and collecting out of databases, out of clinical trial files, out of everything out there that is organized, if you will, into a database is a great example. But you're also pulling what we call unstructured data. So that's not organized. It's not in a database. People have been writing about it. There's abstracts, there's some publications, there might be poster presentations, there's patent applications, like all of that, searching through all of that manually with key terms which oftentimes in software also are not interpreted and used to give you back meaningful information, which is also different about the Lens AI software. You know, being able to do all of that one time and bring all of that information back within the context of omics analysis for target discovery, that's cutting off anywhere from months to years of your research, depending on the software you have access to and the amount of literature research that needs to be performed. When it turns that data back, you have access to all the structured and unstructured data, which is analyzed through natural learning processes or natural language processes, which is really not just pulling back the relevant data quickly, but it's more relevant data because the way it searches is very specific for bringing back information that is very relevant to the language that you used. So there's a lot of great examples. We probably don't have time to go through it today, but there's great examples of how, keyword searches tend to pick up and bring back non-meaningful data outputs, whereas the software is able to cut through that and to really make sure that that output is meaningful. And then it's all there in front of you. You're able to click and search and move through the information that's been brought back. And what is probably, one of the coolest things is because it's not searching entire sequences. You're not getting back all that unrelated information pertaining to the sequences that were not at all relevant to the functional or disease part or target portion of what you're analyzing. And because it isn't searching entire sequences, you're also getting back information that you probably never would have found because it deduced it to a fingerprint. You never would have known that fingerprint was the key association, maybe between two molecules, maybe between a molecule and a disease state, maybe between all the patients that survived a particular disease. You would never know. You just wouldn't know to search for it. And going back to how I love the open minds of the founders at BioStrand, I think that's why the software, I think that's how they were able to create something like this software, because this software requires you to have an open mind. When you get that data back, there are correlations that you never would have been able to find using any other software. And that's really quite a mind-blowing piece, but that's because you're searching a different way. You're searching with different information, and that information is more relevant and more meaningful.
Yeah, no, I think that's one of the big takeaways I'm getting from this is that it's software that a lot of it sort of thinks outside the box, and it's not just that it's sort of you find uncorrelated things that you wouldn't have thought about if you sort of think the old-fashioned way. So it's quite interesting that way. It tells us a little bit just where can the software go? Like what can we expect to see software like this be able to do for us in the future?
Well, so I'll tell you about the few of the things that, because I mentioned there's a lot of things that it can do right now. And there's a lot of things that we believe it's going to be able to do in the shorter term. But maybe I'll kind of go out there and just mention a few of the things that we think it might be able to do in the longer term. And these are, these are the unknowns, right? We've got probably 10 stages before that of like, we can do it now, we can do it in the next couple months, we can do this in 3 or 4 months or 4 months. Now I've got some really big ideas. And one of them I would be willing to even put down as a milestone for us this fiscal year. And that one is what we are already referring to as next century sequencing. So that's taking next generation sequencing, which we already do at IPA and several other companies do. That's really, really rapid, high throughput sequencing. So figuring out, the genetic sequence of different biological materials very, very rapidly compared to what we used to be able to do with traditional sequencing. Next generation or next century sequencing is taking that next generation sequencing and it's actually using the HIFs as the backbone and the scaffold to be able to generate more functional insight from those genomic sequences. So the sequencing takes you to another level, not only speed, but what does it actually mean when you get that sequence data off? You're able to kind of skip over some of more laborious studies as soon as that data is provided. And then another area we're looking into, and we thoroughly believe this is possible because from a theoretical perspective, it certainly is. We just haven't tried it yet. Is, I mentioned to you, it's really challenging to use humans or anything that's really similar to a human, right? When you're going after a target that's human based. And so, people are oftentimes run with this challenge of, do I use antibodies from humans and then just know that I'm going to have a limited diversity even worse for diversity, do I use animals from a transgenic animal that makes human antibodies? Awesome, because I'm getting human antibodies right away, just like I was with a human. So what I get is what I get, right? What I'm working with early on is that same product I'm going to carry all the way through. And if I have to, I don't have to modify it, so I don't have to worry about it changing, and not working anymore later. But the biggest problem with transgenic animals, which, you hear us talk about, you hear a lot of people talk about them, but they are incredibly limited in genetic diversity, right? You took a very limited amount of human antibody genes and put them into a mouse or a rat. And so the amount of antibody diversity and functional diversity coming from those is fairly limited. And side by side from a traditional animal that is not, genetically modified, you typically don't get anything close to the type of diversity and results you get. So that takes you to what most people do, which is, well, we're going to use a different type of animal, and then we're going to actually convert that sequence to one that's human. Well, all of those have cons. You save several months and you start with your product right away with a transgenic, but you also spend tens of millions of dollars. You typically have milestones and royalties, not always, but then you pay more upfront. And you have a really limited diversity. So it's harder to get a good product out of there. Or you go the human route, and then later you sacrifice potentially your products when you humanize them, and it's going to take a lot more time on the end to humanize. Although humanized services are getting better. We think with this software, we may be able to, and this is my bigger, grandiose plan. There's 10 things we know we can do and we're going to do. We think we might be able to use that software to just bypass this entire problem. And theoretically, it absolutely should be able to do that, because again, we don't care about these entire sequences. We care about the hips, and the hips relate to a structure, and the structure relates to a function. The function is binding. So once we know the structural basis of a HIF, we very well may be able to immediately convert that to a human version of a functional HIF from a product that's in the laboratory. And that would be done directly on the same software. And so, that's our one of our big dreams. So in the meantime, we've kind of got we have these offerings now. People are really excited about them. We have ways to completely change the time frame and the quality of what they come to us to develop a new drug with. In the shorter term, we have these really cool things that clients have come to us and said, no one's been able to do this yet. The software sounds like it might be able to. Can we try? We have these existing capabilities where we don't already offer them, but now we can. And then we have these other things where we say, yep, we're going to be able to do this. We're going to be able to build this. We're going to be able to build this. There's already software being built that's not even available yet for services. Each of those levels, and then some of those bigger dreams that just completely change, the bigger challenges that people have had in such a meaningful way.
I mean, it really sounds to me like this could have some massive impacts on how traditional drug discovery is, is being done right now. This could be industry changing.
That's, I truly believe it will be. I truly believe this is the blockbuster acquisition of the century. But that's also why it took us so long, right? Sifting through these companies. And, we have people who specialize in each of these areas. We had some people on contract, very, very well-known bioinformaticists and data analysts. And we would push through the bioinformatics and the algorithms and through some more challenging concepts and collaborations. And ultimately, that in the end, so many of them were just such small incremental steps, a slightly different version of what someone can already do, or maybe making this process of lead candidate selection a little better, a little more thorough, but nothing that really changed this conversation about where we're going, about how this can be done in a so much more efficient way. And that provides an entire roadmap for the future about how to get everywhere we want to go. It's based on intelligent coding now. It's not based on, more force, more power, more energy, and eventually the quantum computing, which was kind of funny for people to always be throwing that out. It transforms the story in a really meaningful way. Yeah.
So you mentioned you're going to go to PEGS. You mentioned, you've got some clients that are sort of early adopters, but how do you really get the message out? Give us sort of a little bit more granular detail. How do you show this offering to your clients or even companies that aren't clients right now? How do they find out about us?
Yeah, no, that's a good point. So, well, I think did I mention that the member of management will actually be the keynote speaker at PEGS? I think I did.
Yeah,
okay. Yeah. And so really getting that messaging out there, I think there's a few ways. So, right now, as I mentioned, we have some early adopters. We won't really push this messaging more broadly until we hit that PEGS event. Getting that messaging out at PEGS is our primary goal. It really start to get that information flowing to different prospective clients. But we also have to tap into a new set of individuals too, where there's cross communication internally, but those individuals who really understand the bioinformatics and the need for the omic analysis that oftentimes are handing down the target to, the person in charge of drug discovery. And so there's a few different ways that we've already gone about starting to tackle this. We have, first of all, a good understanding of who those decision makers are at companies and who's involved on that bioinformatics side. And it is a different way of targeting, and oftentimes those are much more personalized conversations with members of management. And then in addition to that, with our existing clientele, a lot of it is making it accessible and bite-sized for them to really understand, okay, here's the point that I'm at, or here's what I'm doing, and here's exactly how this is going to make this bigger or faster or better at the end of this particular project. And that's what we've been working on in the marketing, with visuals to show exactly step-by-step what they can bite into. And with a lot of the early adopters too, getting them to agree that data that we produced, whether it was a custom project they came up with or something that we could already offer, that data we've mutually agreed to be able to convert into marketing material. So we will almost immediately have case studies of projects done between IPA and BioStrand for our clients to be able to share with other clients and to be able to demonstrate its capabilities. And the power of that in a meaningful way, right? With hard and fast data, which is exactly how we got the Function First platform to take off. We collected metadata, in that case, for 2 years. And then we crunched it and showed people, and they're like, we basically said, here's us, here's everything we can do, here's the competitors. It's just numbers and stats now. And so that's a large part of how we're looking at approaching this, but also taking into consideration that our client attrition rate is so low. We have a retention rate of over 95%. And those clients tend to be not only loyal, happy clients, but a lot of our onboarding of new clients comes from people spreading word of mouth. And that's something that I think we can really help catch fire with having something that's so distinctive in these offerings, where they can't go somewhere else and get these types of offerings. And so, those are all areas that we're tapping into with our sales team.
And then, is it possible to expect that as you guys are, using BioStrand more for your sort of internal, your Talem assets, can we expect that at some point we get an update saying, hey, this is what BioStrand is able to do for what we're doing inside Talem?
Yeah, that's possible.
Okay.
Yeah, absolutely.
I'm fishing a little bit, yeah.
No, I actually love that idea, Paul, because it's a lot easier than being able to share updates with existing partners. Right now, we have several partners that just don't want updates shared. They're like, no, this is not something we would normally share. We don't want anybody out there knowing where we're at in a project, right? Or any details on it. And when it comes to us as a Talem, as a sole owner of a particular asset, that's a great opportunity for us to be able to showcase exactly what we've done and how it helped us. And almost immediately last week, we also started sharing Talem data with BioStrand, saying, this project, this is where it's at, and they had already already tapped into a couple of those programs, but really starting to feed them mass data sets to be able to see what they can do. So, twofold, it improves the quality of those Talem programs. It gives BioStrand, hands-on experience in doing exactly what is important in this type of drug discovery, but also because it's information owned by Talem, the sequences are owned by Talem, it gives us the ability to, not only test out some of these new things and see how they enhance the program, but convert it also to marketing data and case studies. We would just redact the information that would be a potential liability. And anything that might be what we would call an early disclosure. So it doesn't have any risk associated for our patent applications. And, we use that to go out and show the people the power of those capabilities. So
absolutely, I would assume Talem is the earliest of early adopters for this service, right?
Yeah, we could not wait. I think we handed over a ton of different lead candidates, and it's so fun, so fun for BioStrand too, to be able to have access to that sort of information and so many data points around that sort of information, So yeah, it was fun. It'll be great.
Fantastic. Jennifer, I mean, we've covered so much of the BioStrand piece here, but what are a few other milestones we can look forward to sort of over the coming year?
Yeah. So milestones, we've been talking about those a lot lately because we're about to wrap up our fiscal year in like less than 2 weeks. And again, for some of you who might not be as familiar with IPA, we are actually just closing out our 2022 fiscal year. And so starting May 1st, we'll start our fiscal 2023 year. And but yeah, as far as a few milestones we're looking forward to in the shorter term, the first one is our first $5 million quarter. Gosh darn it, we're gonna get there soon. We have to. But things are just going so well, and that's been a small milestone, but it's meaningful because it's we just continue to trend in the right direction. And we've been talking about how our pipeline continues to look really good, and so we're really focused on meeting that milestone in the near future here. And then in addition to that, one of the things that we've talked about for the last couple of quarters, and we've been working on in the sense that we invested in and built out a brand new vivarium facility in Victoria, British Columbia. And it does all sorts of stuff for us. So I won't get into all of that. But as far as a milestone, we're really looking now to develop and offer additional preclinical services there, like efficacy of particular therapies to protect against or treat diseases, PK and tox studies that can be done to assess the safety of different antibodies. Those are not capabilities that we presently have. And they start to kind of gear us up also for the ability to do that type of work in a GLP or good laboratory practice format. And why that's different and why that's important is we can do all those studies, for people at this new facility, and that keeps them with us longer. We've developed the drug, right? They're ready to go on and see, is it truly successful in these animals? Is it really stopping? I'm just going to keep with my cancer example. Is it really stopping the cancer from spreading, doing what we think it's going to do? That's kind of that last step, right? Before you go, right, well, it's working. We're ready to go into a human. That's just another way for us to keep clients with us and to keep quite frankly, spending money with us and keep that relationship going. But the very next step after that is, moving into, GLP or GMP manufacturing. It's a manufacturing level that is used to create a product that has more quality control around it. So that product is made a certain way and you have all the details about it. And the ability to take a higher quality product and use in the same animals with those same protocols means that we would be eligible to provide the IND enabling studies to regulatory bodies so that they can make a judgment on whether the product goes to the clinic. And that's not a capability that most CROs have. and would be a wonderful addition forward. And in addition to that, with the GLP and the ability to tie that into those animal services for IND enabling studies, we are looking to move more meaningfully into the GMP manufacturing services too. So that extends our capabilities beyond research and development and into what's called the CDMO space, which is clinical manufacturing, building the product, making the final product that goes into the human. So those are some of the key milestones that we're looking forward to here in the next couple quarters. Really, meaningful progress in the preclinical and clinical manufacturing, preclinical trials. And then, as I mentioned previously, but I'd also consider it a milestone, that next century sequencing, where we take that next gen sequencing and then it utilizes the HIPs and generates functional insights from those genomic sequences. That really, I believe, would be truly transformative in terms of people's ability to collect massive sequence data that automatically has meaning. So I gave you kind of four goals there that I think are pretty reasonable, shorter term goals. And then as we're really kind of wrapping up this fiscal year and looking forward to the next year, that starts in 2 weeks, we'll continue to provide some milestones for people to watch for and look forward to.
So I'm assuming what you really are trying to tell me is the next couple of weeks or after the fiscal year end, we may get some other updates that you're not privy to say right now. Okay, fair enough. Clearly, everybody's pretty interested in hearing how the talent pipeline is coming along. So I'm assuming when you have some data, you'll make it public for everybody. Is that correct?
We will. So what we're going to be looking to do, we just have so much information right now and so many things going on. And, we'll, as we wrap up our fiscal year end, what we really don't want to do is kind of wait to the AGM time to give updates. So we are going to plan some interim publicly announced calls where we will bring everybody in to give updates, both on our Q4, but also updates of what's happening in Talem and some more updates on BioStrand as well.
Fantastic. Okay, well, we're sort of nearing the end here, and what we always like to do is just give you an opportunity to sort of give a key takeaway or parting message, or cover anything we may have missed. But more importantly, what's the message you want everybody to walk away with today?
A message to walk away with. Well, I think the first one would be, we started off with this vision, right? Like 4 or 5 years ago. And that vision, we knew where we needed to go and we knew where we wanted to be. And I always said, we want to bring the most transformative technologies into this company. And they're not always what I think the mass public thinks what they are, right? I think oftentimes what's out there requires much more than just listening to the chatter, but it's that ear to the ground with the right groups and the right people and really getting in and digging deep about what's meaningful and what's transformative. And, we set out to do that quite some time ago. And it's so interesting how our vision, we were able to kind of provide pieces of it as we go, because we have to keep our cards a little bit close to our chest and not tip off our competition. But our vision, while it does tend to get a little clearer and crystallize as we pinpoint, oh yeah, that is it. That's what we've been looking for instance, with BioStrand, that vision hasn't changed. And what's really changed for us is, for those of you who have been with us on this journey, we spent years rebuilding, this company and structuring it in a way that it was solid foundationally, and it gave me access to the data that I needed at my fingers, and the finance team access to the information they needed globally at their fingertips, for us to be able to ensure that we could really have solid insights and visuals into financials, into sciences, into the way we run things, and get the right people in the right seats, and rebuild in almost every way you can possibly imagine, from restructuring every site, some more significantly than others, building a new site for controlling all of the overseeing of back office and global activities, streamlining the CROs to get them to the point where they are today, where they are all profitable. All the CRO business itself, in and of itself, is profitable. And our spend and our loss is related to building assets, building value add assets in Talem. And then, of course, our dual listing up onto the NASDAQ to get us to the point where, we had some of the funding we needed to move forward. And this whole journey has really brought us to where we are today. Where in this last, 6 to 8 months, we have really been able to put more focus into making that vision happen, and really starting to execute on a lot of these milestones that we've been pointing to, as a vision, as a broader, longer term vision for the company over the last year to year and a half, and really execute on those. And gosh darn it, I think execute really well with BioStrand and what we've brought forward here. And we're so excited to see these pieces falling together. I shouldn't say falling, because it kind of indicates entropy, and this has been a ton of energy, but coming together in a way to build this company where we have this vision in our head still, and we're still just partway there, but that vision continues to crystallize, and that vision continues to become more and more in reach as we continue to execute on these strategies. So I think I said it in the beginning of this fiscal year, we are so excited about this fiscal year and getting to a point where, we raise the money, we have the foundation, we can start executing on these strategies. Now we've done that. And now this year for us is even more exciting. It's a challenging market. These are challenging times with a lot of unknowns happening. And I think we're swimming against the current of the market, but also against the current of where most of our competition is going, which is the wrong direction. And we're embracing very cautiously, but embracing, what we believe we will be able to accomplish this year. And I think we're even more excited than last year. So, that take home is that I think just that pure excitement and that pure belief that our vision really, really will be something that everyone looks back on and says, oh my gosh, that was it. That made sense. That changed the course of history for life sciences.
I love to end it on changing the history of life sciences. I think that's fantastic. I agree with you. I'm very excited about the sort of a new phase that the company's entering right now. I know you've been working very hard to get it positioned for this type of opportunity. And I couldn't be more excited than where you guys are positioned right now, and agree. A lot of things to look forward to as we sort of embark on a new fiscal year. So I want to personally thank you for all the hard work you're putting in, Jennifer, and certainly want to thank you for joining us today. We have been speaking with Dr. Jennifer Bath, the CEO of ImmunoPrecise Antibodies, symbol IPA on the Venture Exchange and IPA on NASDAQ. One last time, I want to thank you, Jennifer, and certainly look forward to catching up with you, and good luck as we end the fiscal year here.
Thank you, Paul. It was absolutely my pleasure. Thanks for having me on.
You got it.